Happy morning to everyone. I welcome you all for the first day of 10 days state level student development program on advances in microbial sciences organized by the Department of Microbiology, Sri Nehru Mahavidyalaya College of Arts and Science, Coimbatore. Prayer doesn't change things, it changes us. Let us start our program by praying the Almighty with prayer song. To shine your brightest light is to be who you truly are. Now we may have lighting of Kuttuvalakku virtually. Thank you. Now I invite Dr. R. Bakiraj, Head Department of Microbiology, to formally welcome the gathering.
Great morning to all of you. It's an opportune time for me to welcome all of you at this gracious occasion. Yes, the Department of Microbiology, Sri Nehru Mahavidyalaya College of Arts and Science, initiated the 10 days students development program on advances in microbial sciences. So the main focus is to enlighten the young minds in the field of microbiology. So it's my pleasure to welcome our chief guest and the resource person of the day, Dr. P. Agila Devi, Assistant Professor, National Pulse Research Center, Vamban, Pudukottai, Tamil Nadu. Welcome, ma'am. I welcome our management members for the program, Dr. Ramesh C. Bafna, the President, Sri Bharat Kumar Jagmani, Secretary, and all the members of our management. And I very much delighted to welcome our dynamic leader, our beloved principal, Dr. Bojan Subramani sir. Welcome you, sir.
Thank
And
you. Thank
yes, sir. And I will cordially welcome Dr. Manjula Suresh, Dean Languages, Dr. Krishna Kumar, Director MBA, and all the heads of the departments and staff members and students, mainly the participants from various institutions across
Hello.
the state. I
Hello.
once again all of you. Thank you.
Thank you, sir. I request our beloved principal, Dr. B. Subramani, Sri Nehru Mahavidyalaya College of Arts and Science, to deliver the presidential address.
The technical team, there is an interruption between the organizers. Please unmute the remaining audio and video. I request all the technical members to mute the mic and listen to the words. And very warm, pleasant morning to one and all present in this Connect student development program, which is going to be there for 10 days. So when we discuss in the forum innovations, creativity, and whatnot. So in that innovation, the national level student development program, it has come out very well, and it was nicely organized by the Department of Microbiology, Professor Bakiraj, Dr. Bakiraj, and the faculty members who are in microbiology. They have organized such a wonderful 10 days FD student development program. So this student development program, it's a common forum. It's a platform created to different kinds of students from various places, various regions. So that gives the technological updations, the academic developments, and some areas, some unique, different kind of understanding level. So when we put in a common forum, so all the students will get a lot of very good informative through this 10 days student development program. And this was focused for all these science stream students throughout the nation, whoever is interested. When any student sharing an innovative thought for the subject, for the domain, that will help any students in the region. And again, it is an ignition point for doing students research and other kind of development, kind of precise study on latest developments in the particular subjects. So that is, I congratulate the entire department. And meantime, I thank our management for giving us the support for doing any kind of academic related programs in the campus, virtually or indirect also. So on all behalf, I thank all the members who connect here, and I thank the resource person for the day, Dr. P. Agila Devi, Professor Plant Pathology, National Pulses Research Center, Vambam, Pudukottai. So we sincerely thank Madam for accepting our invitation and addressing our young minds, not only the SNMB students, throughout the nation, throughout the state, whoever is interested to take up this SDP. So my sincere request here for all the members who are connected, please get the informative. When the 10 days of programs are over, when you consolidate, you should have a very good banking knowledge on advances in microbiological sciences. So that is what expected, out of which any new research innovations, points where the areas to be developed, or points where the curriculum to be developed or enhanced, or kind of practical exposure to be given. So all those things can be discussed in this forum. And when you get a consolidated knowledge updation, that is the success of this student development program. So I wish this SDP going to be a great success, useful for the entire students community, and the organizing team have taken a very good innovative, the SDP as we are there to help the students in all the areas where they are looking actually during this pandemic. So wish you all the best. Have a wonderful 10 days of student development program. Wish you all the best. Thank you for the opportunity given, and I thank all the organizers and the technical team behind this successful, going to be the successful student development program. Thank you very much.
Thank you so much, sir. Personality has power to uplift everything. Now, I invite Ms. M. Sharmila, Assistant Professor, Department of Microbiology, to introduce our chief guest.
Good morning, one and all. I have immense pleasure to welcome Dr. Akila Devi. The management and our department are honored and grateful, ma'am, for admitting our invitation and for making your valuable time efficient in this occasion. Dr. Akila Devi is working as Assistant Professor in Plant Pathology in National Pulses Research Center in Tamil Nadu Agriculture University, Pudukkottai. She pursued as Senior Research Fellow for 1 year. She completed her PhD in plant pathology on bioefficacy, phytotoxicity evaluation, and residue studies of UPF 809 against major diseases of chili. She did her MS research on eco-friendly management of sunflower leaf blight incited by Alternaria helianthi. She is well versed in biochemistry, proteomics, and molecular biology. She was awarded Dr. R. Samiyappan Award for the best thesis work in molecular plant pathology. She holds membership in professional bodies like Journal of Mycology and Plant Pathology, Valarum Velanmai, Coimbatore, Indian Phytopathological Society, and so on. She had published a book, Kalan Valarpu Muregal. She is well known for her popular article on genomics, a developing horizon for plant pathogen interaction, and other articles like mycovirus in plant disease management, applications of scanning electron microscope, applications of nanoparticles, and so on. It is great moment for us to have such great personality along with us. I once again welcome you, ma'am. Thank you.
Thank you, Sharmila. I request our esteemed chief guest, Dr. P. Akila Devi, Assistant Professor, National Pulses Research Center, Pudukkottai, to present on proteomics and genomics. Ahila ma'am. Sir,
Ma'am, whether it is audible, ma'am?
Yes, ma'am.
Ma'am, can I present now?
Yes, ma'am. Please, ma'am.
Okay, okay. So a very good morning to all. Very good opportunity have been given for me for on proteomics and genomics. Before entering into the subject proteomics and genomics, I have to give some brief introduction about proteins. The proteins are the biological molecules or the macromolecules consisting of one or more chains of amino acids. In simple, the proteins are consisting of many amino acids. Okay. Then what are the proteins? The proteins are used for the biological system, and they are mainly used for constructing and maintaining the cells in the plant system as well as human beings. Okay. Then there are certain genes which codes for proteins. The proteins are polymers of amino acids. So what does the protein do? Means it regulates the enzymes, hormones, storage proteins, transport proteins, structural proteins, protective proteins, and toxin proteins. So these proteins are majorly involved in both plants and human beings for their development activities. So protein structure, coming to the primary structure, the primary structure is a specific amino acids which containing one polypeptide chain. And coming to secondary structure of proteins, they are having the primary polypeptide chain, which are folded in the form of alpha, beta sheets, which form forms coils and turns. So the secondary structure looks like coils and turns. And coming to proteomics, we are dealing about now proteomics. The proteomics was the term was first coined by Mark Wilkins. Okay, then what is genomics, transcriptomics, proteomics, and metabolomics? Genomics means the study of DNA. Transcriptomics is study of RNA. Proteomics is a study of proteins, and metabolomics is a study of metabolites, which are essential for plants and human beings. Hello. Then coming to why we are going for proteomics. So the DNA is converted into RNA, then RNA to mRNA, then finally the mRNA is converted into protein. So the behavior of gene products is difficult to predict the gene sequence. Alone with gene products, we cannot see the expression. So if you want to find some expression of a gene, so proteomic study has to be conducted for expression as well as post-translation modification of a cells has to be detailedly studied in proteomics. Then classification of proteomics. Proteomics is classified into categories. One is structural proteomics, functional proteomics, and expressional proteomics. In case of structural proteomics, you can see the 3D structure of the protein. Example, for viewing the 3D structure, example, X-ray crystallography and NMR spectroscopy is used for seeing the structural proteins. In case of functional proteins, we have to see the localization of the proteins and function of each protein in the human system as well as the plant system. And in case of expression proteomics in the plant samples and as well as the human samples. The human samples includes the blood samples or tissue samples, etc. So this is the overview steps which are performed in the proteomic analysis. Yes, ma'am.
மேம் ப்ரெசென்ட் பண்ணியிருக்கீங்களா ஸ்க்ரீனு?
மேம் ப்ரெசென்ட் பண்ணியிருக்கேன் மேம்.
இன்னும் வியூ ஆகல மேம். எதுல மேம் ப்ரெசென்ட் பண்ணியிருக்கீங்க? நீங்க தான் தெரியுறீங்க.
இவ்வளவு நேரம் தெரியலீங்களா?
இல்ல மேம், அதனால தான் மேம் இன்டரப்ட் பண்ணேன்.
ஐயோ சொல்லிக்கலாம்லங்க மேடம்.
நீங்க அதான் இன்ட்ரோடக்ஷன் கொடுக்குறீங்கன்னு வெயிட் பண்ணோம். நோ
ஐயோ
ப்ராப்ளம்.
நான் பாதி ஸ்லைடு முடிச்சிட்டேன்.
அதுதான் மேம். சரி, எப்படி இன்டரப்ட் பண்ணலாம்னு வெயிட் பண்ணிட்டு இருந்தோம்.
முன்னாடி தெரியுதான்னு கேட்டேன். சரி ஓகே. இப்ப தெரியுதா?
ஒன் செகண்ட் மேம்.
ஹலோ.
எஸ் மேம், எஸ் மேம், எஸ் மேம்.
மேடம் இப்ப தெரியுதா?
தெரியுதுங்க மேம், தெரியுதுங்க
சரி
மேம்.
இப்ப ஃபர்ஸ்ட் ஸ்லைட்ல இருந்து போட்டுமா?
ஒரு ஜஸ்ட் மட்டும் கொடுங்க மேம். எங்க தியரிஸ் எல்லாம் லிசன் பண்ணிட்டு தான் இருந்தோம்.
ஓகே. இப்ப
ஸ்கிரீன்
நான்
மட்டும் ஒரு அவுட்லைன்
ஃபர்தரா
மட்டும்
இல்ல இப்ப ஃபர்தரா நான் நெக்ஸ்ட் ஸ்லைட்ல இருந்து போறேன். சரிங்களா?
ஓகே மேம், ஓகே மேம், ஓகே மேம்.
ஓகே ரைட். ஓகேப்பா.
தேங்க் யூ மேம்.
ஓகே. இப்ப வியூ ஆகுலமா? ஹலோ. சரி.
Hello. Hello.
ஹலோ. போங்க சார். இது என்னது? ஹலோ. ஹலோ.
எஸ் மேம்.
மேம் எங்களுக்கு ஸ்கிரீன் தெரியுதா உங்களுக்கு?
ஸ்கிரீன் தெரியுது மேம். தெரியுதுங்க மேம், தெரியுதுங்க மேம். எஸ் மேம்.
ஓவர்வியூ ஸ்டெப்ஸ் ஆஃப் புரோட்டியோமிக் அனாலிசிஸ் தெரியுதா?
தெரியுதுங்க மேம், தெரியுதுங்க மேம்.
ஓகேமா, ஓகேடா, ஓகே.
ஓகே மேம்.
Proteomic analysis, we should take the protein mixture sample. Then protein mixture sample should be converted into individual proteins, and individual proteins are converted to peptides by trypsin digestion. And the peptides are analyzed through mass spectroscopy, that is MALDI TOF. Then we go for protein identification. identification. So coming to the procedure for proteomics, first we have to separate the protein. The separation of protein is done by 2D gel electrophoresis, and after the separation of the proteins, we have to analysis the proteins for mass spectrometry. And this is the two-dimensional gel electrophoresis. For conducting the two-dimensional gel electrophoresis, we have to use the isoelectric point. The isoelectric point is used to separate the particular protein from the mixture of protein. So, depending upon the pH gradient, proteins, the charged proteins moves through the gel until it reaches the isoelectric point. After reaching the isoelectric point, the protein does not move. So then coming for the methods of protein identification, we have to do SDS, that is SDS page analysis for protein identification, and then we have to take the gel and we have to do protease digestion for converting these proteins into peptides. After the conversion of the peptides, they have to be analyzed through GCMS. The GCMS used for the protein identification is MALDI EOF. After analysis of the peptides, the fingerprints has to be taken. That fingerprint is called as peptide mass fingerprinting. After taking the fingerprint of the peptides, then protein identification through software has to be done for finding the structure of the protein and the function of the protein, which is essential for disease identification in both humans and plants. This is the experimental workflow for proteomics. First one is the sample collection, that is it may be a sample from human beings or it may be a sample from the plant. After collection of the sample, it has to be processed. After the processing of sample, the protein has to be concentrated, and the concentrated protein should be separated by 2D gel electrophoresis. After separation of the proteins, it has to be digested, and the digested proteins are converted into peptides. After conversion of the peptides, it is identified through GCMS or LCMS. After that, the protein has to be identified through software, that is Sequest and Mascot software for identifying the function of the proteins. So this is the mass spectrometer. Once the sample is taken from the plants or human beings, the samples are made to bombard with ion source. The ion source may be from electron ionization, chemical ionization, fast atom bombardment, and electrospray ionization, or MALDI, that is matrix assisted laser desorption ionization. After the samples are treated with ionization source, the samples are converted into ions. So after the separation of the ions are separated by quadrupole, magnetic deflection type, electrostatic type, time of flight, and ion trap. After the conversion of the ions are detected through detector. The detector is electron multiplier, multi-channel plate, and Faraday cup. These are the ion detection techniques for identifying the proteins, which after conversion into ions. So how do the mass spectrometer works? So the sample from the it may be from the human sample or it may be from the plant sample which are affected with the disease. Okay. After the samples are taken, it is converted into ions. After the conversion of ions, the ions are analyzed through mass analyzer. After after the analyzing the mass, they are detected in the detector. So this is a simple pictorial form for proteomic analysis. Here the sample enters. After the sample enter into the GCMS, the samples are broken into ion by electron source, and after the conversion of the sample into ions, the ions are detected under detector by field. After the separation of the protein fingerprinting has to be done. What is fingerprinting? The fingerprinting is called as peptide mass fingerprinting. fingerprinting or peptide mapping. This is used for identification of proteins. The separate what I am coming to tell is the separated proteins obtained from the gel has to be treated with the proteolytic enzyme, which is converted into smaller peptides, and the peptides are analyzed for protein fingerprinting for their expression studies. This is the simple pictorial diagram for a peptide mask fingerprint. So this is the gel shows the separation of protein. After the separation of protein, the gel has to be taken and treated with the trypsin. After treating with the trypsin, the sample peptide is analyzed for expression studies. So these are the database available for finding out the function of the proteins. Example, SwissProt, it is a database of annotated protein sequence. It contains what I am coming to tell is the SwissProt is the database which contains many protein database, which is from human sample and plant sample. So we can compare the proteins which we have found some protein. That protein should be compared with already existing database protein, and then we have to come into a result that what is the protein we have isolated is. Then another one is TREMBL. It is a computer annotated supplemented Swiss prot that contains all the translational nucleotide sequence. And PIR is the international unit which contains annotated database of protein sequence, and NCBA translated gene bank DNA sequence, and ESD the expressed sequence tag database, and it's another one is UniProt. It is a database which contain a newly released protein, not released proteins, identified proteins for identification. So proteomic applications, it is used for proteome mining, protein profiling, post-translational modification, protein-protein interaction, and protein network mapping, and protein quantification or differential analysis. Then coming to genomics, genomics is a larger area for both humans and plants. The genomics is a study of interaction between host, pathogen, disease. After studying these interaction, we have to come for gene expression studies. For genomic study, we have to genomic is a study of genomes in an organism. To determine the genome, DNA sequence, we have to study the heterosis, epistasis, and pleiotrophy, which are exhibited by genes inside the plant system and the human system. And types of genomics, there are two types of genomics. One is the functional genomics and structural genomics. Functional genomics studies with the function of a gene in a plant or humans. Structural genomics is a study of entire genome of an organism. It may be fungi, it may be bacteria, it may be virus, or any other organisms. Then why to study the fungal genomics? The fungal genomics which enter into the plant system or human system, it creates lots of varied changes leading to diseases. For finding out their expression studies, we have to genome for genomic study. What are the main area is pathogen metabolism. For finding out the pathogen metabolism may be fungus, virus, or bacteria. What they do inside the plant system, or what they do in the human system. So first one is that one. Second one is cellular and subcellular differentiation by pathogen. After the pathogen entry into the human system or plant system, what are the cellular changes or cellular differentiation exhibited by the pathogen? Then after the studying the differentiation, the pathogen causes signal transduction. After studying the signal translation, we have to study for molecular events. Molecular events is nothing but the genomic study. And finally, molecular events in compatible and incompatible interaction. Compatible interaction means positive interaction pathogen and the plant system. Incompatible interaction may be maybe from the disease and the plant system. And genomics in plant pathology, why we are going for genomics in plant pathology is to study the pathogenicity gene. So when the plant is infected with disease, what is the gene altered by the uh, pathogen? Then coming to the repetitive gene sequencing, what are the additional gene changes which occur in the plant system? And other one is the telomere gene, which get alters after the infection of the disease. Before going into genomics, we have to study gene annotation, microsynteny, scaffolding, coverage, and contig. What is contig? Contig is nothing but it is a sequencing of, it is a short sequence which is used to identify the whole genome sequencing results. And scaffolding is a term used for finding the sequence which is at the end of the whole genome. And the scaffold consisting of many contigs and gaps, to find the length of the sequence in which order the bases are known, we are going for scaffolding. And coverage, what is coverage? Coverage is nothing but the average number of reads representing a given nucleotide. The nucleotide is, we all already aware of the nucleotides. The nucleotide may be adenine, guanine, cytosine, and threonine. So the average number of repeats or reads, by this we have to analyze the genomic changes in the plants as well as human system. And coming to microsynteny is the localization of the gene in the or in the chromosome. Then gene annotation. What is gene annotation? Annotation is nothing but identifying the gene coding for a sequence and gene structure and ORF. ORF is the open reading frame. It consists of three nucleotides. Three nucleotides are used for studying the expression of the gene. And then what is the host pathogen interaction? When the pathogen affects the host, okay, the host may be human or it may be a plant system. After interaction, the signals are passed to the nucleus and the expression of the signals takes place. After the expression of the genes, the important genes for maybe for resistance, maybe for susceptibility. For the resistance, the important genes are MAPK gene, CAMP gene, G protein gene, and ABC transporter genes are essential for resistance in plants for building up the immunity. So these are the functional classification of the gene. The genes which are present in the humans or plants, they some uniformity between the genes present in the plant system and animal system or human system. The genes are responsible for metabolism, energy, cell growth and division, transcription, protein synthesis, cellular transport, cellular communication, ion homeostasis, and cellular organization. Some genes are involved in the pathogen development in plants and human beings. They causes the disruption of the gene or mutation. When a pathogen infects the plant system or the human system, it causes the disruption of the gene and it causes the mutation. After getting the gene mutation has been knocked out. After gene knocked out, the virulence of the pathogen has been increased for their disease. And morphogenesis in the pathogen. The morphogenesis means changes in the pathogen. So variability in the pathogen causes the diseases in humans or in pathogens in plant system. So what are the mutant in genomics? How to study the mutant in the genomics is by four techniques. One is signature tagged mutagenesis, targeted mutagenesis, and restriction enzyme resistance, and transposon arrayed gene knockouts. So by these four techniques, we can find out the mutants in the genomics. And coming to finally, we are coming to the signature tagged mutagenesis. It is used to identify the transposon. What is transposon means? They are the sequence of DNA that move around the different position in the genome of a single cell, which causes the mutation in the genome. And for studying the expression studies because of gene level changes, we have for studying the expression studies, we have to consider the healthy plant and the diseased plant, and the difference between the healthy plants and diseased plant is studied by the expression of the mRNA which is got from the DNA. For studying the expression analysis, for proteomics we go for 2D gel electrophoresis. For genomic level changes we go for EST, that is expressed sequence tag and microarray technique for finding out the um, expression studies. Thank you one and all for giving this opportunity to share my experience in proteomics and genomics. Thank you, sir.
Thank you so much, ma'am. Students can post your queries in chat box if you have any clarifications. Ma'am, I have one question, ma'am. Ahila Devi, ma'am.
Yes, ma'am.
Ma'am, I have a question, ma'am.
Yes, ma'am.
Uh, can you say me, ma'am, some advanced techniques in proteomics to implement for this COVID-19?
Ma'am, actually, we are doing some research on plant pathogen interaction alone, ma'am. We are not dealing about the human pathogen, especially virus, ma'am. We are dealing with some virus which are associated with the plants alone. So,
Okay, ma'am. Please state some advanced techniques in dealing with plant pathogens, ma'am. Recent
advanced techniques
techniques.
in ma'am, for identification of proteins associated with disease, it may be for humans or it may be for plants. We are going for 2D gel electrophoresis for finding out the interaction between the plant pathogen and pathogen interaction, and some after application of a biocontrol agent for plant disease management. Then what is the expressional changes in the plant system? We are studying in proteomics, ma'am. For genomic analysis, we are going for expressed sequence tag analysis and whole genome sequencing for seeing the expression of the genes after the infection of a pathogen. The pathogen may be fungi, bacteria, or virus. Ma'am, can you
Yes, ma'am.
ma'am, then ma'am, is there any other doubts, ma'am?
Ma'am, here are some questions we have received, ma'am.
Okay.
Have genomics, how it can be implemented in human genome?
Ma'am, in human genome, the same techniques, what are the techniques I have told for plants? The same technique only they are using in human also. One is the whole genome sequencing, and then another one is the next generation sequencing. Another one is the expressed sequence tag analysis and microarray analysis for seeing the interaction between the host and the pathogens. Hello. Hello.
Yes, ma'am. Ma'am, one more question, ma'am.
Yes,
We have
ma'am.
received these questions through students. Shall I proceed,
Okay,
ma'am?
okay. Yes, ma'am. Yes, ma'am.
What are the tools we used for proteomic study?
Ma'am, tools is nothing but the 2D gel electrophoresis, and now we are getting 3D gel electrophoresis. In our station, these are not available. These are available at Tamil Nadu Agriculture University, Department of Pathology, Microbiology, and Biotechnology, ma'am. So we can go for 2D gel electrophoresis. That is a recent technology for finding out the protein-protein interaction, then expression of proteins.
Yes, ma'am. Next, we have what are the advantages of proteomics?
The proteomics advantages is for finding out the expression of proteins. So if a plant or a human system is affected by the disease, so after infection by fungi, bacteria, or virus, some proteins are expressed due to the infection of the uh, pathogens. So we can find out the difference between the normal proteins which are available in the human system and the infected plant system or human system to study the expression studies. Can compare those two healthy and infected samples by the expression of the proteins. So some proteins will be expressed in the diseased plants. Some proteins will be expressed in the normal plants. So comparing the sequence or the proteins, that is peptides, by peptide mass fingerprinting, we can find the difference. This is the important application of proteomics in disease management, both in plants and. Hello. Hello,
Yes, ma'am.
ma'am.
Yes, ma'am. Yes, ma'am.
Uh,
Shall I go with the next
is there
question,
any
ma'am?
questions, ma'am?
Yes, ma'am.
Yes, ma'am.
What is the connection in proteomics between biology and chemistry? A student from
Ma'am,
post graduation, she has
regarding
asked
the
this.
uh, see, without chemistry, there is no biotechnology. So, before entering into proteomics or genomics, we have to study the individual nature of the carbohydrates, proteins, fats. After studying those, that is proteins, fats, that in detailed manner, we can proceed for proteomics. Before entering into proteomics, we should have the basic knowledge about the, structural organization of carbohydrates, fats, proteins, minerals, vitamins, everything. So there is a relationship between the, genomics or proteomics with chemistry. Without chemistry, we cannot go into the this technologies.
Yes, ma'am. Next question is,
Ma'am, is there any
shall I proceed, ma'am?
yes, ma'am. Yes, ma'am.
Yes, ma'am. Oh, what is the signature tagged mutagenesis? How it is affected in genomics?
Ma'am, signature tagged mutagenesis is nothing but when a pathogen which infects the plant system or human system, some changes take place gene level. For finding out it may be a mutation, it may be a deletion, it may be addition. For finding out those changes, we are going for signature tagged mutagenesis for finding out the changes after the infection of the pathogen, both in plants or human beings.
Next one question, ma'am.
Hello.
Ah,
Yes, ma'am.
what is the software used for 2D gel electrophoresis? A student asked this.
For identifying the function, ma'am, for finding out the function of the protein, we are going for mask card peptide fingerprinting and mask card peptide software for identifying the protein function. These databases are available in the net. We can compare the isolated protein or expression protein with available database in the net. Hello.
Next one, ma'am. What is the main applications of protein fingerprinting?
Ma'am, not an application. It is a process, and in proteomic analysis, it is a final step. That is, the proteins are converted into peptides by trypsin cleavage. After conversion of the the peptides are analysis for fingerprinting to know the sequence of the proteins. Uh, it is a complex it is converted into simple as peptide. After the conversion of the peptide, we go for sequencing of proteins for identification of their functions and their expression.
Yes, ma'am. Next one. What are some practical applications of proteomic research?
Proteomic analysis is used for identification of disease-resistant protein or disease-susceptible proteins and expression proteins after the fungal attack or virus attack or bacterial infection. We can study the proteomic expression.
One last question, ma'am.
Yes, ma'am.
How the proteomics can involved in hairy root culture?
Ma'am, actually, I'm not involved. I'm not aware of those things.
Okay, ma'am.
Harry root
Okay,
culture.
ma'am. No issues, ma'am. Because students are posting the questions, so that's why I've asked, ma'am.
Ma'am,
No issues, ma'am.
you just give me. Okay, okay, okay.
Okay, ma'am.
Thank you, ma'am. Thank you.
Okay. Thanks for
Thank you.
the active participation from students. Now, I request Mrs. M.B. Kavita, Assistant Professor, Department of Microbiology, to propose the vote of thanks.
Greetings from Department of Microbiology. It's my privilege to propose vote of thanks on this gracious occasion. I, on behalf of the Department of Microbiology, SNV CAS, first of all extend my most sincere thanks to the Almighty. It's my pleasure to extend a very hearty thanks to the chief guest, Dr. P. Akila Devi, Assistant Professor, National Pulses Research Center, Pudukkottai, Tamil Nadu, who spared time from her busiest schedule to grace the occasion. Thank you, ma'am. I'm glad to extend my sincere thanks to the management for their support for organizing this program. I must mention my deepest thanks to our principal, Dr. B. Subramani, for his sincere motivation and support. Thank you, sir. Let me take this opportunity to thank Dr. R. Bhagyaraj, Head Department of Microbiology, for his encouragement and dedication. Thank you, sir. My sincere thanks to all the heads of the department and staff members of various colleges and SNV CAS for their support. My hearty thanks to the technical team, office staff, non-teaching staff for their support. Lastly, my sincere thanks to the student community who are joined with us today. Thank you all.
Thank you, Kavita ma'am. Thank you, Akila ma'am, for your thought-provoking address. Thank you so much for your time, ma'am. Akila ma'am. Thank you all for all the students. Kindly subscribe the YouTube channel MB Online. And tomorrow we'll have a second day of our student development program at 10 o'clock. Thank you all.